version of animals, an early history of animals on Friday. Probably this weekend, maybe Friday night, I will post a worksheet, a video worksheet for you guys to do. I still have to decide how, because it's a two-part video, and so I'm kind of decided if I want to do one or both parts. But it's pretty good, I think so. Anyway, that'll be there'll be something. I just have to decide exactly which version of the worksheet that video I want to use. But that'll be posted probably Friday, I would guess, Friday after lecture, after lab. So Friday night probably. So look for that, and that will be then due like a week later or so. Oops, and we are, there we go. All right. So anyway, get on to, and of course lab is whatever lab is. Flatworms and other weird little critters. So more microscope work in lab. All right, and just a reminder. I think on Wednesday, next Monday, I think is I'll have a sign up for whatever projects you want to do, so I can get an idea of how many people are interested in each. So you might want to be there for that. All right. Anyway. So today we're going to look at the protozoans. We've already covered some of these in lab, just to kind of give you a little bit of a feel for the diversity of this group. But it's a very, very basic look at a group that has hundreds, tens of thousands of species or more. I forget exactly how many different protists there are. So the old kingdom Protista, which really is not a monophyletic group, and just really isn't used in formal classification anymore. But the old kingdom Protista basically is all of those eukaryotic organisms that are fairly simple in their structure, that are not plants, fungi, or animals, essentially. And so these generally don't, many of them don't have true tissues, although some of the marine algae, multicellular algae, the seaweeds, might be sort of an exception to that. But unicellularity, or fairly simple colonies, is pretty common in this group. But at the sort of biochemical level, they're incredibly diverse at the cellular level. So some of them are heterotrophic, some are autotrophic. They differ in the kinds of molecules they use to store energy, whether it's glycogen or cellulose or something else. And so many of these are not particularly closely related. They're really just at the base of the eukaryotic tree, all of the various branches. So this is clearly a paraphyletic group, and could easily be split into multiple kingdoms. Before we get into some of the members of that group, I want to talk a little bit about the origin of eukaryotes in general, which is basically the origin of protists. You've probably covered, I presume you've covered this sort of in intro bio, at least in a basic sense, and that is a serial endosymbiont theory. So basically what this serial endosymbiont theory says is that eukaryotes originated from a prokaryotic ancestor. The actual main cell probably is derived based on DNA sequence studies from a member of the Archaea group of prokaryotes. And from there, you have some sort of an ancestral nucleus. How the nucleus formed is actually up for debate. There are a couple of different theories, whether it's an infolding of the plasma membrane, whether it results from the fusion of separate archaea cells, or even an outfolding, so that the original plasma membrane essentially corresponds to what is now the nucleus of a eukaryotic cell, and the rest of the membrane system is kind of an outfolding of the original plasma membrane. So there's a lot of debate about how the nucleus evolved. We're not going to get into that. But then what we do have much better evidence for is the origin of a number of key organelles. The first of these that probably evolved is the mitochondrion, which evolved through endosymbiosis, which basically means the retention of another cell internally, endo, and so both were mutually benefited each other. And that other cell would have been a heterotrophic prokaryote that used a highly folded membrane to basically create proton gradients that it could use to generate energy much more efficiently than cells that only relied on simple glycolysis, which only gets, 2 ATPs per glucose molecule, something like that, while a aerobic respiration extracts a lot more energy and uses oxygen as a final electron acceptor. And so of course, by engulfing this bacterium, basically engulfing it via phagocytosis, but instead of digesting it, that bacterium was kept inside of a vesicle. So now you have the bacterium cell, you have the membrane around it, and over time, and because that bacterium was able to generate a lot more energy, it was retained, and some of that energy was used by the host cell, and the host cell in turn provided useful resources for the bacterium. Over time, that bacterium became much more simplified. It lost many of its genes, not all of them, but many of its genes and some of its genes were actually transferred into the nucleus as it became more and more reliant on its host, and essentially became an organelle in its host, the mitochondrion. So mitochondria essentially derived from an ancestral bacterium. What the endosymbiont theory, serial endosymbiont theory suggests has actually happened multiple times after the origin of the mitochondrion. The next one is the origin of the flagellum, which is now believed to actually have originated from a type of spirochete bacteria. This is a bacterium that has a greatly elongated, spiral-shaped body that it can flex. And so that bacterium was basically incorporated into the host cell. And again, all of the non-essential bits of the bacterium, its own DNA and its own ribosomes and things like that, were basically gotten rid of. And just the cytoskeletal elements that allowed it to flex were retained as a flagellum. So there's now reasonable biochemical evidence to suggest that flagella are actually of endosymbiotic origin as well. They are originated from a bacterium. And then cilia would have been just modified flagella. They have the same internal structure. They're just more shortened and coordinated and so on. But probably a flagellum was the first version of this. And then finally, at least one group of early, so this is the origin of your basic eukaryotic design. That is, you have a flagellum, which could then be modified into cilia and things like that. You have mitochondria to be aerobic. And so this basic cell gave rise to many different lineages of eukaryotes. But at least one lineage of these then underwent another case of endosymbiosis, in this case picking up a photosynthetic prokaryotic, in particular a cyanobacterium. Cyanobacteria are bacteria that are capable of photosynthesis, and one type of cyanobacterium was absorbed into a host cell, and again, just like with the mitochondria, it was largely reduced to retain just the photosynthetic properties. Many of the other basic maintenances, ribosomes and things like that, were all lost and taken over by the host cell, so that it essentially became just an organelle within it. And that, of course, gave rise to chloroplasts specifically. But chloroplasts can lose, in some cases, actually lost their photosynthetic function, but were still retained for other things. And we'll see one example later on. And so the term plastid is used more generally for any sort of chloroplast-derived structure. Chloroplasts themselves are plastids, but you can also have non-photosynthetic plastids as well that arose from the same thing. Now, if you look at the phylogeny of eukaryotes, one of the things you'll see is that the photosynthetic groups are actually scattered, let me jump ahead here. I'm going to find that picture. Maybe I'll have to. Yeah, here we go. Here's a phylogenetic tree. So the major photosynthetic groups include the red algae and the green algae. Because they're closely related to each other, even though they tend to have different accessory pigments that gives them generally different color, they probably represent a single origin of photosynthesis here. But there's also another group here, the stromatophiles. These are the brown algae, the kelps and things like that, as well as the single-celled diatoms and so on. Most of the guys here are also photosynthetic. And so that suggests that photosynthesis evolved in this group here. And then there are scattered photosynthetic groups in some of these others as well. Let me see if I can find, there's euglenids, for example, which include about 30% of euglenid species are photosynthetic. All of these, and there may be a few others scattered here and there, almost all of the other photosynthetic groups, besides, I think the brown algae are the original, I hope that's right, but I'm fairly sure the brown algae represent basically the sort of the first origin of chloroplasts within eukaryotes. And all of the other photosynthetic eukaryotes basically are derived from another level of endosymbiosis, where basically a brown algae was captured by an ancestor of these guys, for example, and its chloroplasts retained for those groups. The same with euglenids, which are derived, whose chloroplasts are derived from a green algae. And so you have sort of secondary endosymbiosis of chloroplasts happening as well. There was actually only molecular data shows there was actually only a single origin of eukaryotic chloroplasts from cyanobacteria, and then the other groups basically picked up their chloroplasts from a ancestral brown algae or some sort of ancestral spermatophile. It's also worth noting that the origin of chloroplasts here probably actually predates the split between the alveolates and the spermatophiles, and probably occurred here because a number of species in this group actually have plastids, but are not photosynthetic. They seem to have lost their photosynthetic abilities. All right, so I think that covers that slide now. Questions so far? All right, so we're not going to talk about all of the protists. That could really be an entire class on its own. But we are all of the the protists. So I'll just give you a quick overview here. We can kind of group them into three sort of ecomorphological types, three basic ways of making a living. We have the photosynthetic protists, the algae and so on. Algae can be single-celled or multi-celled. The multi-celled would, of course, be the seaweeds. It's unicellular algae, often referred to as phytoplankton, in that they're basically cruising up in the water column in the plankton and so on. And so these are all of the photosynthetic group, or most of the photosynthetic groups. You have a bunch of sort of heterotrophic protists that are essentially fungus-like. They mostly digest external to the cells. They secrete digestive enzymes and then absorb nutrients. And some of them form long filaments and things that look a lot like fungi. So things like water molds and slime molds have traditionally been viewed as being fungus-related. They're not necessarily particularly closely related to the true fungi, but they're basic fungus-like ones. And then the third group are the protozoans, which are the ones we looked at in lab. These are the most animal-like in that they tend to be they're again heterotrophs, but they tend, like animals, to be somewhat ingestive in that most of the food is processed internally within a vesicle, basically, so the food is engulfed through phagocytosis or some similar means, and then digested within the cell. They tend to often be relatively mobile, so they then can have that similarity to animals. So, and protozoan, the name zoa means animal, and so proto means first or before or early. So protozoan basically kind of is the old idea that these are the ancestors to animals. But of course, these three none of these form a monophyletic group. These are polyphyletic. We have, algae on very different branches of the eukaryotic tree. Fungus-like protists are kind of scattered about as well. And then there's a lot of things that don't fit into these. These are kind of simplified categories. There's a lot of things that don't fit into them. Like, what do you do with a euglena? It can consume external food like any other protozoans, and it's closely related to non-photosynthetic groups. But it's also algae-like in that it has chloroplasts and can photosynthesize. So there are lots of critters here that don't necessarily fit into these groups. So don't take these don't take these too seriously here. It's just kind of a way of summarizing the kinds of diversity we have amongst, protists or eukaryotes that aren't plant animals or fungi. All right, and so many of the various protist groups or phyla have things that can be called protozoans, and many of the protozoans used to be kind of classified based on their morphology, on like whether they had cilia or flagella or pseudopods and things like that. Much of that has kind of fallen by the wayside, molecular data in particular, some microstructure looking at how the cells are put together essentially at the molecular level, cytoskeletal elements and things like that, as well as in particular DNA sequence data, suggests that many of our traditional views of how these were related are completely wrong, and things like flagella are probably pretty much ancestral to them and are just retained in various groups. Things like pseudopods may have evolved independently multiple times, or maybe also was an ancestral feature. We don't really know. All right, so this is kind of I think I showed you the same slide, or I think it was the exact same slide, in lab when I introduced the protozoan lab. So I don't really expect you to kind of memorize this whole thing here in detail, but it does show the main clades, some of the main clades that are typically characterized by animals we think of as protozoans, that is, the more mobile, heterotrophic groups here. So the clade alveolata, which is a sister group to a group that includes the brown algae and diatoms. Remember I showed you those are the ones that might also have, probably ancestrally, perhaps had chloroplasts. Includes the ciliates, the ciliophora, the dinoflagellates, and the apicomplexa. So the ones, and let's see how, I don't know how well those colors look in the scrappy screen, but the ones in purple, that includes the ciliophora, are ones that we looked at in lab and I'm going to talk about here. The ones in red are ones that we're going to be in lecture. The ones in blue are the ones that you've seen in lab, but I'm not going to talk about here. So the ones in blue you're not responsible for in lecture at all. Those we covered in lab. The ones in red or in purple are ones that we do. So some like ciliophora, and I'll mention the radiolaria a little bit. Briefly, those we'll cover in lecture, and we already saw them in lab. And the choanoflat, and then the other guys in red here are ones we didn't see in lab at all, but I'm going to talk about with. Since this is an animal quest, the choanoflagellates being kind of the most important in terms of origins of animals. But we're going to start up at the top with the clade alveolata. That means that is, this could be roughly considered sort of a kingdom level grouping. That includes a diverse array of ciliated, flagellated, and non-motile protozoans here. The molecular data also are related to each other. So we'll start with this, and so I already showed you this. So we are starting with the alveolates, so the mustard yellow group here. They're related to the spermatopiles, which we won't look at because these are mostly algae-like things. And then we're going to touch upon the amoebas, and we saw some of these guys in lab. And then this is the cons here. This is the clade that includes the fungi and a few protozoan groups related to it, the animals, and also the choanoflagellates, which are the sister group to the animals. All right. So anyway, let's start with the phylum Ciliophora. It's a reasonably good group, big group, about 8,000 species, relatively diverse in their structure and in the kinds of lifestyles they have. When you have that many species, that's kind of to be expected. What characterizes them is the presence of cilia, these are the major. This is the major cilia-bearing group. They've evolved cilia. They've kind of converted their ancestral flagellum into the cilia. So cilia have the same basic structure as flagella. They're generally shorter, but beneath the plasma membrane, there's a much more complex cytoskeletal structure that allows them to be linked together, so that the cilia can be their activities, their swimming motions, can basically be coordinated across the body. Flagella are not coordinated in that same way, so they're in a way a more advanced kind of feature. So some cases the cilia may cover most of the cell. This is a paramecium, scanning electron micrograph of a paramecium. This is, you guys saw it, what is it? You guys remember? I'm blanking on it. I'm asking you because I can't remember. We had it in lab, so, but I forget. Anyway, and there's the little labels here. Anyway, this one has cilia. It has free cilia, but it also has masses of cilia that kind of fuse together into a larger structure that can actually be applied pressure to substrates and so on, so they can almost kind of walk across the surface. And so sometimes they may be entirely covered in cilia. Other times the cilia are more restricted to certain regions. Those cilia tend to make them pretty fast, and they're among the fastest of protozoans. And all of the ciliophora have a pellicle, which is underneath the plasma membrane, basically a proteinaceous layer underneath that helps them give them that three-dimensional shape. There is actually quite a bit of variation. So here's Vorticella, a nice little video clip here showing the spinning cilia are form a little ring here. The mouth would be at the in the center of that funnel, and these guys are actually attached via the stalk here to a substrate. So they're relatively sessile, and they bring food into them. But many ciliates, of course, are mobile, and in fact, these guys can swim as well. They can detach, and then the cilia actually just draws them through the water. So they are actually capable of swimming, but most of the time they prefer to be attached to the substrate and bring food into them. While many of them are free living, there are some species that live inside of other organisms. There are gut endosymbionts that digest cellulose, for example. So found in termites and other organisms. Some ciliates actually produce an external test. So this is actually the test here. So this is secreted external to them. They can kind of withdraw into that. And so that's something that's evolved independently in a few ciliates are actually parasitic as well, mostly on fishes. Although parasitic lifestyle isn't particularly common in this group. Okay. They have a fairly diverse set of reproductive strategies, and often reproduction in this group is very complex. So complex, and we're not going to actually go into it because I don't really want to spend 15 minutes trying to explain paramecium sex to you. But they can reproduce both sexually and asexually. Most ciliates have two kinds of nuclei. They have a large macronucleus, which is polyploid, that contains many copies of their chromosomes, multiple homologous sets, maybe potentially dozens of them. And then they have a number of smaller micronuclei. They may have one or more, or 1 to 4 micronuclei. The micronuclei are normally diploid, and those are the nuclei that are primarily used for reproduction. The macronucleus seems to contain most of the genetic instructions that the cell uses in its day-to-day life. The micronuclei are used in reproduction. In acellular reproduction, that's what the picture here shows. The paramecium just undergoes binary fission. It splits into two. The micronuclei undergo meiosis. The macronucleus just splits into two, and each daughter nucleus gets about half of those, half of the DNA, and then that DNA, those chromosomes can be replicated. Sexual reproduction, which I didn't put in the picture for because it actually requires a whole series of pictures, basically involves conjugation, where the bacteria connect together and with a tube that allows them to exchange micronuclei. Prior to conjugation, those micronuclei undergo meiosis to produce haploid micronuclei, and then usually half of that micronuclei get transferred from one paramecium to the other, and the other is, so if you send out 2 micronuclei, you also get 2 micronuclei back, and then those micronuclei can fuse together to form macronuclei. And meanwhile, fuse together to form a diploid so those haploid micronuclei fuse together to form diploid nuclei. But they all then undergo mitosis to form a new macronucleus, which is dissolved in the meantime. So it's a whole really complicated process here as well. But the basic point that I want you to kind of keep in mind is that the micronuclei are basically used for conjugation, for exchange with another individual. It's a form of sexual reproduction, basically. Each resulting cell now has a different genetic makeup than it had before. And so they're used for sexual reproduction, while the macronucleus is basically just an ordinary sort of cell runner, organizer, basically. So these guys can reproduce both sexually and asexually, yes.
So is that micronuclei a form of like plasmid?
Huh?
Is the micronuclei a form of like a plasmid? Their form of plasmid?
No, so it is a eukaryotic nucleus, so it can have multiple chromosomes. And those chromosomes are linear, just like in other eukaryotes. They're not circular like a plasmid would be, like a bacterial plasmid. So they're basically a nucleus. So the difference between a micronucleus and a macronucleus is that macronucleus just has crap loads more DNA in it, because it's basically, but it has essentially so a diploid micronucleus is going to have multiple, so I don't, and I suspect the haploid number varies among different species of paramecium and different ciliates. It's going to have, let's say you have 3 different homologous chromosomes. So a diploid micronucleus is going to have 6. So if one set has 3 chromosomes, a diploid micronucleus would have 6 chromosomes total. Just like any ordinary eukaryotic cell would. These are eukaryotes. And then it undergoes meiosis, just like other eukaryotes do, to form the haploid ones. It's the haploid ones that are actually exchanged. And then they can fuse together, the nuclei can fuse together to form new diploid micronuclei. And a macronucleus is just basically take a bunch of those, a little more complicated than that, but basically take a bunch of those micronuclei, shove them together, and have a polyploid nucleus with multiple homologous sets of chromosomes. But they're exchanged, yeah, so I mean, in the sense that they're exchanged like bacteria exchange plasmids, there's conjugation involved, but the mechanics of it are very different, because you're not just exchanging a small piece of DNA, you're actually exchanging an entire nucleus as they do so. All right, that's all I'm going to say about it. You saw ciliates, and you got at least a taste of their diversity in lab. And remember, this is an animal class, not a protozoan class, so we're going to move on to, where are we on time here, okay? Our next phylum is the dinoflagellates. I think your book calls them the dinozoa. There is actually technically a difference between dinozoa and dinoflagellata, and I forget which one. So dinozoa is actually a slightly larger group that includes a few obscure species, dinoflagellates plus a few other species of the dinozoa. But dinoflagellata is a much more familiar term, which is why I prefer to use it. So anyway, estimates are somewhere between 2,000 and 4,000 species. The big range here is because in a lot of cases they have fairly complex life cycles. So when you find 2 different dinoflagellates, it's possible they may actually belong to the same species and just be different stages in their life cycle, if you don't know the whole life cycle for these guys. And so our lack of understanding of some of the complexity of life cycles means that we also don't know for sure how many different species there actually are. So the name dino, di meaning 2, and flagellata, flagella, basically describes one key feature. They have 2 flagella. Those flagella are basically at more or less right angles to each other. So one fits inside of an equatorial groove that wraps around the cell this way, around sort of the equator of the cell. So it's the flagellum sits in a groove, and it sort of undulates within that groove and produces water currents and causes itself to spin. And then there's also a longitudinal flagellum that also sits in a groove here. It actually extends beyond it in this species. But there's also a lengthwise groove that contains a flagellum that in some cases may extend beyond that. They also have a pellicle, but it's actually a rather different structure, and it's much tougher in most species than in the ciliophorates. It's actually, and it's fairly armored, that is, there are a series of vesicles beneath. And actually, it says beneath the plasma membrane, but it's actually a little more complicated than that, in that they essentially have two membranes, two plasma membranes, and so the vesicles sit between those two membranes. They are filled with cellulose typically, and they act kind of like an armor plating. And with decent microscopes, you can actually see those individual plates on many of the species. And so that helps maintain their shape. They tend to be much more rigid. Most of these are much more rigid shaped than the ciliates, some of which, like you saw in lab, can actually be fairly flexible. Their pellicle is not as nearly as rigid, much comprehensive. Most dinoflagellates are unicellular. There are some that form small colonies and things like that, but they're by and large unicellular. And about half of the species are photosynthetic, and tend to be either reddish or brown. Those that are photosynthetic tend to be fairly reddish or brownish in appearance due to the accessory pigments associated with chlorophyll. Chlorophyll is green, but it's the accessory pigments help absorb stray light, either capture additional light or protect from too much light. It's those accessory pigments that tend to give them their reddish or brownish color. And about half of the species tend to be photosynthetic through secondary endosymbiosis, where probably capturing chloroplasts from brown algae. Okay. This one here, this is, oh, I don't have the name up here. That's, I forget what the name is, but anyway, this is a common freshwater photosynthetic species. All right, so here's some other looking one. Here's some other forms. This one's like, you can see the kind of groove right here. That's where the circumferential flagellum fits here. And again, here there's a notch here, and here represents the equatorial groove for the one flagellum. And then here's a longitudinal flagellum out behind it. And again, you can see some of those armor plates here as well. So in terms of their life cycle, they are able to reproduce, just about all of these guys can reproduce asexually, by simply fissioning. That's an ancestral character, so basic cell division, fissioning is an ancestral character for protists as a whole, for eukaryotes as a whole, or for pretty much life on Earth as a whole. Many of them can produce sexually as well. They seem to they seem to be mostly haploid. They will then fuse together to form a diploid zygote that then undergoes meiosis. So that's the form of sexual reproduction. Basically, you join with somebody else, combine your DNA, and then split again. One interesting feature here is that they are known for carrying large amounts of DNA, typically about 5 to 10 times the amount that's normal for most eukaryotic cells. Typically, they're, so you can see here, this is actually the nucleus here. The nucleus is often enormous. It can make up to half the volume of the cell as a whole. But that DNA is generally condensed. It does not unravel except during DNA replication. It doesn't have the histones that organize the DNA like we get in other eukaryotes. And so why they have, so that also means that most of the since it's basically permanently condensed and doesn't have the histones that might, mark it for parts of it for not reading. We don't really know what all that DNA does, if it does anything, or why they've evolved this. It appears to be a unique feature of the dinoflagellates. It's a large amount of DNA that isn't actually, that's basically condensed, only briefly uncondensed during DNA replication. But even during cell division, it retains, through mitosis and so on, it remains in its condensed state. Okay. So one of the reasons I talked about dinoflagellates at a little bit more length than we did with ciliates is because they're actually pretty important ecologically, especially to animals. So many species are plankton. The majority of species are marine. They are freshwater forms. In fact, This is a freshwater form here. We've actually used it in the past in the lab, but because that lab always tended to run long, I dropped it, and because they often didn't survive very well, so it was hard to get good specimens that weren't just dead things sitting at the bottom. So anyway, but the majority of species are marine. They tend to be found up in the plankton. Many species, so I said about half are photosynthetic, but they're in fact probably mostly mixotrophic rather than obligate or complete photosynthetic or autotrophic organisms. That is, most of those that have chloroplasts can actually switch between heterotrophic and autotrophic modes. They can shut down their chloroplasts and mostly consume other organisms or organic material. Other times they can switch and become mostly photosynthetic. So they're pretty versatile in their lifestyle. Many of them are also bioluminescent as well, so that's another feature. So we get like glowing tides and so on, glowing water. It's almost certainly going to be dinoflagellates in that water. And they're also capable of very massive reproductive blooms. So their populations, because they're kind of versatile, if there are lots of nutrients available, for example, lots of nitrogen and so on, then they can essentially populate. They can switch to autotrophy. They don't have to worry about feeding anything, and their populations can undergo massive population booms, which we call blooms or red tides because they're often red colored, and they can literally stain the water red. They've been recorded in nature in densities up to 100 million cells per liter. So about, think of a quart bottle. That would contain about 100 million cells of these guys. And they seem to do particularly well when there's excess nutrients like nitrogen in the water. And so at least some of these massive population blooms are associated with human waste runoff. So excess nutrients due to fertilizer runoff, due to livestock waste, due to soil erosion, which brings nutrients into the oceans, and so on, that can often trigger these blooms, both naturally and a lot of times through human effects. There were big popular blooms of red algae following one of the big hurricanes about a decade ago or so. That hurricane, which hit the Carolinas, basically flooded a lot of pig farms there, and a lot of the pig farm waste got basically washed up with the withdrawing floodwaters, washed out into the ocean. So lots of big influx of nutrients led to subsequent red tides in those off the shore. Okay, so those red tides are actually significant to humans and other animals because many of these dinoflagellates not only photosynthesize, but many of them are also quite toxic as well. Some of them produce very powerful neurotoxins. Carlodinium, for example, is a species that produces toxins that basically immobilize other algae that it then consumes for food. Mobilizes their flagella. Lots of algae still have flagella, phytoplankton, and they basically disable that so they can capture them. One that's actually gotten some attention in past years is a dinoflagellate called Physteria, Piscicida. It has actually is one of those that has a very complex life cycle. 24 different sort of life stages have been identified. Some are autotrophic, some are heterotrophic. They differ in various things. And some stages are highly toxic. They produce toxins that can weaken fish and open up sores. Those sores mean that the fish is bleeding blood and fluids into the water, which provides food for these guys. And the dead fish, so it causes fish to die. So they've actually led to outbreaks of fish kills. Because it's a neurotoxin, some of that gets vaporized, and fishermen, not even eating a fish, and just working in the water above the fish, have come down to various symptoms, headaches, blurry vision, things like that, when there's a Physteria bloom. So it can be even dangerous just to be in or above the water when there's a big bloom. Many of these dinoflagellates are fed on themselves by filter-feeding, suspension-feeding organisms, like clams, for example. And so when a clam takes in a bunch of dinoflagellates, they're often adapted to tolerate those kinds of toxins, and will actually store those toxins basically sequester them, But what then happens is that humans can come along, eat that clam that's been feeding on toxic dinoflagellates, and suddenly those neurotoxins infect humans, and you get what's called paralytic shellfish poisoning. You're eating clams that ate dinoflagellates, and you can basically take up neurotoxins that cause you to be paralyzed and die. And so that's why there are, why if you live anywhere in the coast, that there's always, seasons or lots of shellfish fisheries might be closed whenever there's a dinoflagellate bloom outbreak, because that's when the shellfish become no longer edible by humans. So they have practical implications. The other really important thing that dinoflagellates do is that, as I said, there are a significant number of photosynthetic species, and those photosynthetic forms often become endosymbionts in various animals and even other protists. So radiolarians, which are an amoeboid-like shell, you guys saw those in lab, for example. So radiolarians are basically heterotrophic themselves, but they can take in dinoflagellates. Now remember, radiolarians are single-celled, so they're actually taking an internal dinoflagellate. Am I out of time? Yeah, I'm out of crap. Well, they can be, let me stop here. I guess we'll get to that one. It was too much of a trip. So let's stop here. We can pick up dinoflagellates next.